Hello, ladies and gentlemen. I'm Daniel Montano. I'm the Chief Executive Officer of Zhittya Genesis Medicine. Today, we're going to give a presentation on Parkinson's disease, medical research study using intranasal delivery of Fgf1. First look at the data. My partner, Dr. Jacobs, is here. He and I will be giving you the presentation. I'm fairly convinced that you're going to like it. Our drug candidate, which is Fgf1, we've been working on this for 23 years. Jack and I have expended in excess of $150 million, and to date, we've had outstanding outcomes in all the applications, the heart, diabetic foot ulcers, and now I believe the brain in humans. This Zoom webinar will be focused mostly on our recent medical research study on treating people with Parkinson's disease. I believe you'll find the medical results very encouraging. Important topic that I'd like to say in advance is that the first candidates, the purpose, we dosed 8 people to find out if the medicine is safe. I believe that you're going to see the evidence, and I believe it's safe. I would also make the comment that we believe that this application could also be applicable to other neurodegenerative diseases, brain disorders, stroke, Alzheimer's disease, multiple sclerosis, traumatic brain injury, on and on and on, because we believe that the vascularization of the brain is a fundamental problem. And I believe if it's safe in the brains to test on people with Parkinson's, it'll be safe in the brain to test on people with strokes, ALS, multiple sclerosis, traumatic brain injuries, etc. We have had success, and Jack will demonstrate our success with the heart. We've had success with diabetic foot ulcers, and I believe now we have human success with Parkinson's disease. Our first medical research study, we have treated 8 subjects with absolutely no adverse events seen or being reported by any subjects. This Zoom webinar will focus on our recent 5-week medical research study that took place in the British Virgin Islands. Dr. Jacobs will show you a video of the motor skill testing on the 2 subjects, and you can make your judgments of improvements for yourself. Now, please understand, we had, as we said, 8 subjects, 2 agreed to be videotaped and to allow us to use their image. Some of the other people did not want people to know they were involved. You'll also see interviews with the 2 subjects and the study's principal investigator, Dr. Harlan Vanderpool of the British Virgin Islands. The first medical research studies that we're launching after, and I hope the August timeframe, we'll be doing 2 additional medical research studies for Parkinson's disease. The first cohort will be dosing on August 1st, which will be for 14 days at low dosage of FGF1, ending on August 15th. The cohort 2 will start on August 17th and will be for 7 days dosing at the high dose. Motor skill testing will be conducted before and after each dosing, and obviously, everybody, we do complete safety checking along the way. We are seeking 4 test subjects for each cohort for a total of 8 people in the month of August. Now, Dr. Jacobs will go over the medical and scientific issues. Dr. Jacobs has been my partner for 24 years. He's a PhD in molecular biology. He was at Washington University in St. Louis when this molecule was discovered. Jack has been with this molecule for in excess of 40 years, even though he's only 23 now.
Thank you.
I will introduce you. Dr. Jacobs, please take over.
Okay, thank you for that kind introduction, Dan. About a half of the viewers on this webinar have not really heard our presentation before, so I just want to bring everyone up to the same speed. So give a little background on the drug we're developing, how it works in our bodies, some past clinical success we've had with this drug, and then spend the bulk of the time looking at the study we just completed in the British Virgin Islands. We'll show you some motor skill testing on the subjects. We'll interview the subjects, and we'll be talking with the principal investigator who ran the study, Dr. Harlan Vanderpool. Okay, so our drug, FGF1, that stands for fibroblast growth factor 1. It's a protein like insulin. It's not a mRNA or gene therapy. It's a natural protein in our body. And it stimulates this process of angiogenesis. So angio is blood vessel, genesis is new. So this drug stimulates the growth of new blood vessels when needed. If you put this drug into normal tissue, not much happens. But if you put it into damaged tissue, ischemic heart tissue, ischemic brain tissue, you see the generation of new blood vessels. And in the brain, that's coupled to new neuron growth as well. The skateboarder scraped his elbow. He's going to use FGF1 to heal that wound. Let's take a little bit closer look at that. So let's say this dotted white circle is the scab on his elbow. Let's look underneath that scab. You can see here brand new blood vessels. These are capillaries that have been formed, stimulated by our drug, FGF1. And what they're going to do is they're going to perfuse this wounded area on his elbow and regenerate skin, whatever he needs, hair follicles, nerves, and regenerate skin as an organ. And so the same process can occur in the heart, and I'll show you also in the brain. And the nice thing about FGF1, it can not only form these smaller vessels, but it can also form small arterioles. So it's really good at getting new blood flow into wounded areas. Here is the FGF1 protein. Dan mentioned I actually was working on this at WashU as a graduate student. We were purifying it from cow brains, and we're not using cow brain FGF1 in our clinical trials. We actually make it. This is the manufacturing facility we use. It's FDA inspected. So we're using industry standard technology to make FGF1, which is pure, potent, and really suitable to use for human studies. Okay, I just want to take a second to talk about our previous work in coronary artery disease. I'll show you a little video on a trial that I supervised in the United States. These are patients with severe heart disease. They can't have a bypass. They can't have a stent put in. Really, this drug was their only hope. They were called no-option heart patients. So what the surgeons did, make a little incision and then inject the FGF1 into ischemic areas of the heart. And we saw new blood vessels grow. We saw them exercising better. And ABC News came to one of our clinical trial sites at the University of Cincinnati and actually interviewed the cardiologist there as well as some of the patients. Let me just run that for you. I won't talk through it, but I'll come back. It's about a minute long. So I'll be back in a minute.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
So these heart studies, there are actually 3 independent heart studies. Over 60 patients were treated. Really gave us a good look at the safety of FGF1. It was safe in humans, and that it really was a potent grower of new blood vessels, at least in these heart disease patients. Okay, I want to move now. First, just give a little preclinical or animal data. We always have to do that before we go into patients. I'm going to show you little data in mice on stroke recovery, where you can really see nicely FGF1 stimulating new blood vessel growth in the brain, and that's coupled to new neuron growth in the brain. And then we'll move on to Parkinson's, where there's data in primates and a monkey model of Parkinson's, again, where the dysfunctional dopamine neurons in those monkeys were regenerated, leading to better movement in the monkeys. And then we'll move on to the human trial that just was completed. Okay, you can give a mouse, this is a human, but in a mouse brain, you can tie off a small middle cerebral artery, so an artery in the brain, and you can give that animal a stroke, and they develop brain damage just like a person. And then you can visualize that, as shown on the next slide. So these are brain slices through a lab mouse. I've said this many times before, if you believe in reincarnation, you don't want to come back as a laboratory mouse. But here's the stroke area right here. This is the white infarcted, this is damaged brain in this whole panel. And then on the right, we have a mouse that received FGF1 intravenously for 2 weeks. So I think you can appreciate the healing, not quite back to normal, but certainly healing. And we can look in these brains and look for new blood vessel formation and then test the mice, are they doing better with their motor skills? So let's look at the vasculature. This is the dense capillaries in the mouse's brain. We're just looking at the blood vessels. Of course, in the brain, there'd be millions and millions of neurons in this area as well. When we give that mouse a stroke, you can see we decimate the blood vessels. This is what causes the brain damage. And then the animals that got treated with our drug, FGF1, you can see a regrowth of blood vessels, not quite back to normal, but certainly better than the placebo-treated animals. This is basically sugar water. These animals are trying to recover, but you can see these vessels formed. These are hemorrhaging. These animals are not doing nearly as well as these animals. And we can infer that we're getting functional neurons by doing behavioral testing. So a very common test is to put these mice on a rotating bar. So this is a rotating bar. Normal mice grab on with their feet, their claws. Here are the stroke mice that were treated with FGF1, hanging on pretty well. And then the placebo-treated animals can't hang on at all. So from this, we can infer we're forming functional new neurons in these mice. So this is ready for testing. We have a clinical protocol, which we've put together, which will we test in humans who have had a stroke, recovered, but have a permanent disability. And we will be treating them with FGF1. Okay, Parkinson's disease. I know a lot of you know a lot about this, but let me just review. So in Parkinson's disease, it's the dopamine producing neurons that become affected. And these dopamine neurons, we have billions of neurons in our brain, and only about 500,000 of them are these dopamine producing neurons. Majority of them are located in a very tiny area of the brain called the substantia nigra. Dopamine neurons are pigmented, so they appear dark. And we and others feel Parkinson's is initiated by a gradual choking off of the blood supply to this area of the brain. So as the blood supply gets blocked and choked off, these dopamine neurons become dysfunctional. So healthy blood flow, healthy neurons, normal movement. In Parkinson's, reduced blood flow, reduced dopamine movement disorders. And you can actually look at blood flow in the brains of normal people and those affected with Parkinson's. And what you can see is shown on this slide. This looks at MRI, functional MRI imaging of blood flow in healthy adults and subjects with Parkinson's. The study looked at a very small area of the brain where the dopamine neurons are housed. This is a healthy 21-year-old, maximal 100% blood flow through the dopamine neuron containing area. We know as we age, even a healthy 65-year-old, we're getting reduced blood flow basically throughout the brain and also in this area where the dopamine neurons are. But in Parkinson's disease, we see almost a 50% reduction in blood flow. And we believe this is what's choking off dopamine neurons in this disease. And if we can restore blood flow and restore new neurons, that we perhaps could reverse Parkinson's disease. So the gold standard for testing drugs before they go into humans is a primate, a monkey model of Parkinson's, where a neurotoxin is injected into the brain of these monkeys. And after about 7 months, they come down with irreversible Parkinson's disease. So let me show you the results on the next slide of testing FGF1 in this monkey model. Let me kind of explain this graph to you. So we're looking at motor scores here. This is a scale. 4 is normal movement, 1 is abnormal movement. So the monkeys get injected with this neurotoxin, and we'll have 2 groups of animals. One in blue are going to get, eventually, later on, are going to get FGF1, and the red group is going to be the placebo group. But first, we have to let both groups develop their experimental Parkinson's disease. So at about month 9, both groups of animals have gait disturbances, they have tremors, they're displaying a lot of the classical motor symptoms of Parkinson's. We then treat one group with FGF1 in blue. These guys still keep getting placebo group, and you can see, immediately we're starting to get separation between the FGF1 treated animals and the placebo treated animals. And again, we're looking at motor skills. So these guys are getting better. Perhaps if we treated longer, they would have gotten back to normal, but there's a real disparity between the FGF1 treated monkeys and the placebo treated monkeys. What we think is going on, so we start the FGF1 here. We think over the first couple of weeks, we're getting angiogenesis, new blood vessel formation, and then that's coupled to neurogenesis or new neuron formation, and we start gradually seeing improvement in motor skills. The dosing here was different from what our patients got in the trial we'll be talking about, but I think where our patients are now are probably in this range, so we're hoping that they'll continue to improve in time. So we can look in the brains of these animals. We can look in that tiny area called the substantia nigra and say, are we growing? Are we regenerating new dopamine neurons? And indeed, we are. The FGF1 treated animals, brown, are brand new dopamine neurons. Here you see a lack of those neurons in the untreated animals. So we believe in monkeys here, we're regenerating dopamine neurons, kind of addressing the root cause of the disease in these animals. Now, in monkeys and also humans, they develop plaques. These are toxic tangles of protein, similar to what you see in Alzheimer's disease. In Parkinson's, they're called alpha-synuclein plaques. And you can see this is a plaque aggregated. This is a toxic kind of tangle of proteins. This is outside the neurons, but this is toxic to the dopamine neurons. And you can see with the FGF1 treated monkeys, we're able to really get rid of most of this plaque. Here's a normal animal. And we believe this is due to reestablishment of blood flow, of good perfusion in these monkeys. So that not only is regenerating new dopamine neurons, but it's getting rid of this toxic plaque. So with this type of data, we are very anxious to get into humans. We submitted a study to our US FDA. They liked the study, but they wanted more animal data, which is always disappointing. We submitted it to Mexico. This was giving the drug intravenously. They approved it. But in the meantime, actually during the pandemic, we stumbled across something which I think is really going to revolutionize how we can deliver our drug for these brain disorders. We were contacted by a group in Minnesota who had pioneered the use of intranasal delivery. They were squirting insulin up the nose of patients with Alzheimer's and doing this in clinical trials in the US. They asked for some of our FGF1, which they made radioactive. And in Minnesota, they squirted this up the nose of animals and then looked to see if our FGF1 could get into the brain. And with insulin, it gets in through two nerves, the olfactory nerve here. This is how we smell. Our nerves penetrate our nasal cavity, but go back into the brain. And this is the trigeminal nerve. So I'll show you on the next slide. Indeed, our FGF1 did get into the brain. We compared it to intravenous dosing, and twice the amount of FGF1 got into the brain versus IV dosing. So let me show you that. This is a busy slide, but I want to make three main points here with this slide. First is FGF was widely distributed all throughout the brain after intranasal delivery. So the researchers in Minnesota looked at different areas. There's a substantia nigra, so this is important for Parkinson's. But they looked at all areas. Here's the hippocampus. Motor cortex is important for ALS. And this is the amount of FGF1 that got in. And then it's in a concentration, which is not important. But the important thing is this is how much gets in intranasally. This is how much gets in intravenously. So almost twice the amount gets in just squirting it up the nose. And we were going to use a central PICC line in our clinical trials, which is a surgical implantation of a catheter into the patients. And to be able to do this intranasally really makes this much simpler and I think a lot safer. Second point I want to make is this red line here. This is the line we know you need to get above that for FGF to have activity in the brain. So if you're below here, you're not going to get new blood vessels forming, you're not going to get new nerves forming, but above this red line, you're going to fully activate these what are called FGF receptors, and you're going to get new blood vessels and new neurons. So really doing it either intranasally or IV, I think we would see a response in people. Okay, so with this in mind, we started a intranasal dosing study in the British Virgin Islands, which is very eager for clinical studies, biotech. They have a very good record on compassionate use types of studies. We teamed up with the doctor who had good relations with the government down there, got approval to do a 5-week clinical study. Daily dosing, we started 2 weeks of low dosing, gave the patients a week off, and then a week of high dosing or double the dose. And as I'll show you, I'm going to show you some videos. The drug was safe in the patients, well tolerated. No one developed bloody noses. And you'll hear our principal investigators talk about any adverse events. And we looked at this very carefully. We did lab testing, looked at people's eyes, looked at their vital signs, EKGs. We saw no adverse events. So as Dan mentioned, FGF1 intranasally is safe in Parkinson's disease patients. Importantly, we were using the U.S. Movement Disorder Society's Parkinson's disease questionnaire, which is kind of the main instrument was used in clinical trials to gauge improvement or non-improvement in motor skill testing. And we saw a 50% improvement in the part 3 of that, which is the motor examination. We'll be looking at some patient videos to kind of document that. I'll give you my impression, my opinion, but you form your opinions. Let me just show you. So this questionnaire, which is used in clinical trials, looks at both non-motor and motor aspects of daily living, but the motor examination is the critical point. This is where the patients actually do standing up from sitting in a chair, touching their nose, tapping their fingers. And this is considered probably the most important aspect of that questionnaire. The lower the number, the better. So a score of 0 means you're completely normal. A score of 4 means you're severely affected. And so the 2 subjects who consented to be interviewed, subject 1, you can see an improvement of 46% on this motor examination. Subject number 2, a 53% improvement. So overall, just with these 2 subjects, we saw about a 50% improvement in these motor skills. So what we're going to do now, we have a video. I'm going to talk through the first 7 minutes, which is basically this motor examination. And then we'll have interviews, a series of interviews that I'll be interviewing both subject 1 and subject 2. They'll be at home now. They went down to the British Virgin Islands for 5 weeks. They've been home for about a month, and we're going to talk to them at their homes. And I think you'll find that very interesting. Subject 2 actually has been to a physical therapist who did some third-party testing to make sure that what we were seeing was real, and we're happy to report that it was. So let me move on to the video. Okay, so we're back in the British Virgin Islands, and we're going to do before. This is before FGF1 treatment, before May 3rd, and then after. This is basically 4 weeks later doing testing. This is subject 1, doing a walking test before and after treatment. Kind of showing his turning here. You can judge for yourself whether you think he looks better or worse after treatment. We did some posture. Drew an orange line before treatment and after treatment. This is subject 2. In the same walking test, this is looking at gait. This is before treatment. It's after treatment. And we'll show you this again, but she's going, in our minds, walking a lot faster, with a lot more confidence after she was treated. She seems to be happy. She's shaking her hips there. She was a great participant. And here she is here, walking a little bit more slowly. Yeah, this is starting at the same time to get a feeling for how fast you could do this walking test. Turning is part of this. We looked at turning. See, her turning before treatment was a little bit slower. There she's coming through the finish line again. And again, she was able to complete the test in a lot less time after treatment than before treatment here. Shows her turning, and her posture, I think, is much better after treatment than before treatment. And we'll look again at her posture with a little orange line. Again, before and after treatment. All right, this is another movement test in part 3 of the questionnaire. Tapping fingers, before, after. It was before, I think, having some problems. After, in my mind, that looks improved. There's subject 2. This is before and after. Before, she was struggling. Her right hand is her weaker hand, so you can see she's struggling there. And this is after the treatment. This is her weak hand. This is looking at hand balance, tremor amplitude, how much your hands shake, and the amount of shaking. So this is before treatment on May 3rd. And here's after treatment, 4 or 5 weeks later. And this is an active movement. She still has some tremor on rest, you can see. But on an active movement, I believe her tremor's really gotten a lot better. This is her bad arm, her right arm. Again, in my mind, in my opinion, a lot steadier. Try it again. We asked them to try to hold it for 10 seconds in this test. Weaker arm here, again, for 10-second hold. Okay, this, excuse me, actually shows the intranasal device we're using. It's a drug, the FGF1 is in this reservoir. This is the electronics, yellow part, you insert it into the nostril, press the button, and so this shows subject number 2 kind of before treatment. This is her first treatment, and then close to her last treatment. And yeah, it takes some dexterity to insert it into the nostril. She could do it herself here, but before treatment, she was really struggling to get it in to her right nostril. Someone had to come and actually steady her tremor, as shown here, where she's kind of doing it all on her own after the treatment. And again, after the treatment there, she's showing off her tremor. She was very cooperative and great attitude. Okay, so this will be the last motor skill we're testing. This is a very standard standing up in a sitting position from a chair 3 times with your arms crossed. Subject 1 after treatment, before treatment. He's struggling a little bit here. He's, in my mind, in my opinion, not struggling here. He's going to try his 3rd time here. He's doing more than 3 on the right, but here he's, again, struggling before treatment. Here's subject number 2, after treatment, struggling, get up. Nice smooth movement. Staying up, okay, again, struggling before treatment. And then she did not even attempt a third treatment. Okay, so we're now going to move to interviews. I'm going to mute myself because you'll hear me talking here. So we'll be talking with patient 1, Daniel. Hello, Daniel. I want to reiterate to our viewers that These patients took part in a 5-week study. Before we gave them any of our drug, intranasal FGF1, we screened them. So we did a screening procedure. You saw that on that 7-minute video. That was the before scenes. They then were treated for 2 weeks, low dose drug, week pause, 1 week of high dose drug, and then we did the additional screening again that you saw in that 7-minute video. That was after. So the before and after. And as we announced a while ago, that the patients improved on the all important part 4, which is the motor examination. You saw them walking and tapping their fingers. But now we just want to interview the patients, more relaxed. They're back home. Questions about safety, how they're feeling, any changes in movement or memory. And for our viewers who don't know, Daniel Clark is a physician. He's an anesthesiologist, and he gives us, no bull. He gives us the true story. So he's a real scientist and a physician. So we appreciate that. So Daniel, I'm going to ask you a series of questions here. Take your time. And so we're going to talk a little bit about safety. So when you're taking the intranasal FGF1, while you were squirting it up your nose, did you have any pain, discomfort, or any other adverse sensations, such as stinging feeling, something like that?
Negative.
Negative. Okay. Now, since you stopped receiving the medicine, since you've been home, have you noticed any adverse feelings in your nasal cavity, in your nose?
No, I did not have any negative sensations in my nostril or a sinus.
Good. Well, let's talk about other areas of your body. So since the first time you took the medicine up until now, have you had any discomfort or pain in any other part of your body that you could believe was related to taking FGF1?
No.
Have you felt any nausea, lightheadedness, or change in your heart rate at any point during the study or after the study?
I have not had any problems.
Now, I know you weren't really affected severely or even mildly severely with tremors. So, but any difference you notice in any type of shaking or tremor activity?
No, I do have a mild tremor when eating, when I get the fork or spoon close to my mouth. That seems to be unchanged at this point in time.
Now, what about when you're walking? So balance when you're walking, any changes in your gait or feelings of imbalance while you're walking?
Balance is about the same. My gait is improved. I noticed that during our walking test that I was able to turn more easily. That may be a small sign that's of improvement.
Okay. Now, I know it's hard for you, but for your posture, I think we saw on that video how it appeared your posture improved after the treatment, before and after.
Yeah, I've become more cognizant of my posture.
Okay.
And that has in turn related to maybe improved gait.
Good. As we've mentioned in the past, our monkeys who were treated with the FGF1 improved, probably over a 7-month period. So, okay, Daniel, have you noticed any changes in drooling or nasal discharge? You said that had been an issue for you before you started the clinical trial. So any changes noticed in that?
Yes, the nasal discharge seems to have improved, and the drooling is maybe slightly improved, but the significant improvement is the nasal discharge.
Oh, that's good. That's good to know. Okay, we're going to switch over to talking about any changes in memory, cognitive abilities. In the literature, it talks about cognitive functions are the slowest to change. So, but just in general, have you noticed any improvement in your memory or cognitive functions from before dosing, before May 3rd to this point in time?
Hard to say.
Okay.
That's difficult to answer. I'm not sure if that's a plus, minus, or about the same.
All right. So you talked about mood, apathy, kind of emotions, trouble focusing, I remember. So have you noticed?
There is a possibility of assessment here. I've noticed that I'm able to pick up a novel and start reading it because I had a difficulty doing that before. I have noticed I used to read in bed a lot, and I've picked up the habit again. So that is something that's enjoyable, and that's a positive thing.
Right. It requires focusing too, right? And you got to read.
It requires focus and concentration.
How would you summarize your personal experience with the FGF1 medication and participating in this trial for Parkinson's disease? Any general comments, anything you would improve, anything you thought worked well? What we learned from you, Daniel, is going to help guide us in the future. So we appreciate your contribution.
Well, and I appreciate you choosing me for this test. And I appreciate the work that all of you have done in this regard, because without the vision of Dan, without your expertise, without Vika and her diligence and going through the literature, this would not have come to pass, because there's nothing really going on in Parkinson's treatment. Levodopa, carbidopa is the mainstay drug, and that's going nowhere. All you do is dose yourself up, watch the side effects, and watch yourself spiral down the drain. So there's no hope. And all of you have given me hope, and you're giving hopefully millions of people hope, and hopefully a cure. So I thank all of you very much.
Thank you. I hope you enjoyed watching your improvement on that video as much as I did. Now I want to kind of ask you more relaxed questions now you're home. Had your friends and families meet with you. So we're going to talk just generally about any adverse sensations or events you've had. We're going to talk if you noticed any changes in your physical movement or motor skill functions. And then we'll get into cognitive memory issues, and then just an overall assessment of how you felt participating in that trial, how you feel now. And of course, we'll hear from Chris after you're done. Let me go through a short list of questions addressing safety and adverse events. So when you were using the intranasal administration device, that yellow device, did you have any pain, discomfort, or other adverse sensations when you're using it?
No discomfort at all.
Okay.
Awkwardness, the tremoring, but that got better.
Yeah, we saw that in the video. Yeah, okay. Now, since you've been home and you haven't been taking that medicine, have you noticed, any problems in your nasal cavity, any soreness or discomfort?
Not at all.
Not at all. Okay. Have you experienced any discomfort or pain in any other part of your body since you've stopped taking the medicine?
No, nothing to do with the medication at all.
Good, good. Anytime since you started taking the medicine, did you experience nausea, lightheadedness, or say changes in your heart rate, feeling your heart speed up?
Nope.
Nope. Okay. I like your answers. Overall, can you think of anything, any bad events, any adverse reactions to taking this medicine?
Not at all. It was really easy to use. There was no issues at all.
Okay, good. Since the first day of dosing, which was May 3rd, until today, which is June 20th, have you noticed any change in your appetite?
No.
Any change in your tremors?
Not. I haven't noticed a big change since we've been home.
Since the beginning, yeah, since May 3rd.
Oh, from the beginning, yeah, there was, yeah.
Yeah, I think we saw that on the video, and
Yes.
we're going to hear a bit more maybe from your physical therapist who you saw after you came home, right?
Right, and I could actually put makeup on again because I could hold my hand by my eye.
Makeup, okay, good.
I couldn't do that before.
Since you started taking the drug on May 3rd, any change in your walking gait? I think we know the answer to that.
Oh, definitely.
We saw, we saw improvement in your posture on the video. Now, one thing our other subject talked about improving was nasal discharge or drooling. Have you noticed any changes since May 3rd in that?
Yeah, I had that definite change in that. It was like a constant drip in my nose to maybe 90% gone.
Wow.
I mean, it was a huge change. And the drooling was probably about 60% better.
Okay. Now, have you noticed any change since you started taking the drug in your sleep patterns?
Oh, definitely. I've been sleeping a lot better.
So,
I used to wake up and I couldn't go back to sleep. I'd be awake for several hours in the middle of the night. Hasn't happened since.
okay.
I would have to go use the little ladies' room, but other than
Okay.
that.
Okay, that's okay. Your activities of daily living, so changes in eating, using utensils, getting dressed, hobbies or other, can you do things now that you couldn't do before you came to the British Virgin Islands?
It's a little bit easier to handle the utensils. I still shake and trimmer, but at least I can hang on to it and I can actually cut food a little bit. Before I couldn't do that either.
Okay. And I think Dan Montana mentioned something about being at church. You could do activities that you couldn't do before.
At church,
Yes.
I haven't been to church for a couple of years. I was just not up to it. I couldn't handle it. When we got back there, I was able to walk and do all the things I normally did, and I was able to sing.
Oh,
Couldn't sing before. My voice would crack and get all full of crud. And I know it's a Parkinson's thing, but it's gone. It's still there a little bit. I mean, I can't sing beautifully yet, but
did your church members turn and look at you and say, well, my goodness, she's singing. That's great. Now let's move to functions that change more slowly in time, memory and cognition. But do you think you've noticed any improvement in your ability to focus or to recall events or just memory in general?
I don't recall ever having problems with that.
Okay.
I don't think there's really a big change in that. Personally,
Okay.
my husband might be able to tell you more, but I don't see a difference.
Okay. Yeah, I think you had more physical limitations than cognitive. I don't know, changes in your mood or emotions, happier, sadder, apathy, more energy.
Well, there was one, yeah, with that there is. I used to take Sammy for depression. That's a little herbal thing that you take. And I weaned myself off of it while we're there at the BVI.
Okay.
I've been fine. I have no more depression.
Wow. Okay, well, that's definitely an improvement. All right, let's bring your husband. Okay, Chris.
Go ahead.
I can ask you, Chris, you want to tell us a little bit about yourself, your role as a caregiver, and then maybe your overall impressions on how your wife is done.
Absolutely. I met this beautiful lady 51 years ago. We got married, and we have 4 beautiful children. We do everything together practically. We do not take separate vacations because she's so adorable. I know that in the 49 years, we were always happy, and it was sad to see her go through depression when she found out she got Parkinson's 13, 14 years ago. Being with her has become, for lack of a better term, chore, because she's so dependent upon doing the things. And as a husband and a loving husband, I don't want anything happening to her. I don't want her to fall. I don't want her to do anything that may cause any problems, and you hear about all those things in life where people fall and everything. And I did have to balance her a lot. Every time we go out and we do something, I'm always holding her hand and steadying her, opening a car door for her and letting her in, because I just don't want her to trip. And even when we were at the BVI, it was more cautious because it was a medical / vacation. So I didn't want anything to happen to her. And actually, which was really surprising, yesterday for Father's Day, she got up and she cooked, and she hasn't done that for years. She hasn't cooked for years. To me, that was a miracle by itself. Just standing up and getting around what I felt was more important. She had a lot more energy than she's had for years. And like don't want to reiterate what she said, but she's sleeping a lot better. She's starting to, I guess, return to normal, if I can use that, or getting more normalized in her life and her living as the woman that she's always wanted to be. So I definitely do see improvements with her. And like what was shown on the video, her walking around here unassisted by me and just being happier, that is a treasure to me, because the last thing that a husband wants to see is his beloved spouse hurting all the time. It's just seeing incredible things happen to her. And like she said, her balance is a lot better. Every once in a while, she gets a little bit off balance, but I would have to say probably at least 80% of her time, she's balanced. Even when she's putting on her makeup, how she keeps her hand steady and she can put on her makeup and her eyelashes and all that other stuff. And so I'm kind of being excluded out of the part of caregiver because she's starting to take care of her own self.
Okay, Chris, we know a lot of Parkinson's disease patients become isolated. They don't want to go out in public, don't want to be seen. And just want to ask you whether you've noticed any change in Becky's activities of that type since she's returned home.
Yes, significant. We go out considerably, a lot actually. And I know in the beginning she's always made a comment, I don't want to go because I don't want to be embarrassed, because she couldn't cut her meat or she couldn't hold her fork straight. And she ended up going from her right hand to her left hand to eat. And she didn't want people watching her eat. In that time, I ended up cutting her meat and to where it was more manageable for her to eat easier. It's not that way anymore. We go out, we eat with friends and family, and she doesn't care who's there. Her shaking has gotten considerably better, so it's not as noticeable. It's not gone yet, but it's not as noticeable, and she's not as embarrassed.
Good. Thank you. Okay, Chris and Becky, you both were in the British Virgin Islands for 5 weeks. Chris, you described it as a medical vacation, but maybe you could give some of our viewers kind of what your overall experience there was in this medical research study and what maybe future patients, future cohorts of patients have to look forward to in the British Virgin Islands.
You want to go first, Hutton? Well, the people were just really friendly. The doctor that was there was very supportive, and they just kind of just took us in. They were really compassionate. We made quite a few friends there. It was surprising. They told us we're family, and we actually did. We felt like family there.
We met them at our church, and they took us right in. It was really wonderful. And one of the young men there named Jermaine saw me, and I talked to him the first day, and I told him what was going on. I told him I said it was Parkinson's trial and stuff. And he told me later, he says he didn't think it was going to work, but afterwards he saw changes in me, and he was like going, "Whoa, this stuff is really amazing." He in the 3 weeks that we were there, that he knew me, he saw changes.
Becky and Chris, since you've returned from the British Virgin Islands, have any of your friends or caregivers or family noticed any differences? Because they really had a 5-week hiatus seeing you, before you were treated, and then 5 weeks of treatment seeing you after the treatment. So if you could share anything in that regard with us.
It was pretty exciting. When we got home, 2 of my sons that picked us up from the airport were there. And one of them came up to me, and right away, the first thing he said was, "Wow, Mom, you're not trembling near as bad as you were." He noticed immediately.
Good.
What do you think?
Oh yeah, they definitely noticed the difference. And that was good, because even as a caregiver, I'm noticing differences each day as she's progressing. And having somebody that's known her all her life notice the immediate differences coming back to the airport was just a confirmation to me that the FGF1 factor that she was taking made a huge difference.
I had my best friend write down her opinion of what she thought was different, because she knew me before and after, and Chris is going to read it out of my journal.
It says, this is Karen Bailey, who's Becky's friend. Becky has noticeably less intense hand tremors and leg movement. She seems in less discomfort in hips and legs, also as she doesn't have to shift positions as often while sitting. She appears to be able to move neck and head more easily and has to stop to reaction balance when crossing the room, much less than before her trip. So in other words, she noticed, and she comes over all the time, she noticed all those changes with Becky the first day when we come back and she was over here. So she noticed those things. And the interesting thing is that my wife has been going to physical therapy for many, many years. And so for the Parkinson's. And the therapist there wrote a couple comments after she was done treating Becky. One of the comments was, her static balance is up 25%.
That was being able to balance on a balance board and keep it level for 30 seconds.
I don't think I could keep it for 30 seconds. And then it says 5 times sit to stand time, dynamic balance, 50% improvement. And that just goes along to all the tests that were done at the BVI.
That's wonderful. Yeah, to have your physical therapist of years do physical testing on you, not
Yeah.
is saying you look like you're better. I mean, you were better. So that confirms our testing as well. That's great news.
I do want to tell you guys one thing, that back 10 years ago when I first had this first two symptoms, and the doctor told me two years later that I had Parkinson's, I had no hope. It was a really sad thing to, and all these people out there have the same problem I had. There was no hope. And then I turned to God and got the impression when I found you guys that you were the ones I need to follow. And you guys have given me hope back and proven proof that it works.
Definitely works.
Yeah.
Thank you.
We thank you for your participation. You were a brave young woman to be one of the first, and we're truly thankful for that. Okay, viewers, I'd like to introduce our principal investigator for our research study at the British Virgin Islands. It's Dr. Harlan Vanderpool, and he's with our patients the entire time. Did a lot of the testing on them, and so I just want to ask him a few questions about the trial down there. So Dr. Vanderpool, during the month the patients were in the British Virgin Islands, what is your general opinion on how they responded to their treatment?
Well, Jack, I thought that they responded really, really well to their treatments. They were very positive in their approach to their treatments, and I could see remarkable results and improvement at the end of the study.
It was good to hear. So Dr. Vanderpool, did the patients report to you during the course of their treatment any event you would consider an adverse event related to the drug they were receiving?
None whatsoever. We did not receive any reports of any adverse events. I didn't observe any adverse events at all with both subjects. So that went really, really well. Okay, and we did eye exams, and I think you administered the eye exams, and how did they look? That's correct, Jack. We did eye examinations using standard fundoscopy, and we found no adverse effects on the vasculature of the eyes. Both subjects had normal vasculature when we started and remained normal throughout the period of time. Good. I had a chance to review the laboratory values that you sent me, and from my reading of the values, they looked fine. They stayed within normal limits before, during treatment, and after treatment. So in my mind, that looked pretty good. I don't know if you have any comments on that. Nothing at all, Jack. I thought those laboratory results were normal throughout the entire study, and no issues at all. Now, to your knowledge, was there any change in the patient's vital signs after they received the drug? So, we looked at their vital signs, I think 5 or 10 minutes after getting the drug. Yes, Jack. Vital signs were stable before administration of the drug and also after administration of the drug. No adverse effects in terms of vital signs. Okay. Noted. Did you see anything we may change? We're going to be bringing additional cohorts of patients down to the BVI, and we're going to try different dosing regimens. But other than that, did you see anything that you might want to change in our next research study with you? Well, I just, we could continue to collect as much data as possible. I believe that the monitoring things like the vitals are so critical, but I believe that we've demonstrated very clearly the marked improvement the patients have seen, and it was an awesome experience. Yeah, I think the video does not lie. You can see it with your own eyes, and you saw it with your own eyes, and so we're very pleased with the outcome. Okay, so just in final wrap up, I mean, anything good or bad that you want to say about the study? Or sounds like you were pleased with the conduct of the study, with the outcome, and I think look forward to doing additional patients in the BVI. Yes, Jack, I was absolutely pleased and thrilled with the results of the testing. And I believe that our subjects were also equally pleased. Daniel and Becky were great persons, awesome individuals, and really happy for them just to see their dramatic improvement. So I was over the moon, as Dan says. I was completely over the moon. Great. Okay. Dr. Vanderpool, thank you for your comments. That's all the questions I had.
Okay, we're going to come back here. We're going to wrap it up now. We have a few more slides. As Dan mentioned, we have the clinic in the BVI, but we also are in negotiations for clinics in other countries, including the Bahamas, Panama, Cayman Islands, Albania, and Morocco. So we hope to have many clinics up and going, doing these very focused research studies. Dan laid out what we're going to do in August in the British Virgin Islands, looking at 2 additional cohorts of Parkinson's patients. One will get a low dose, second cohort will get a high dose, but we also have additional studies planned with Parkinson's disease. For example, a cohort of patients that have DBS, deep brain stimulation, and want to be treated with the FGF1. Parkinson's patients who had DBS and had it taken out, who wanted to be treated with FGF1. So we're going to be doing a number of subsets of Parkinson's disease patients, but also there's promising animal results and literature results that suggest that FGF1 could be potentially beneficial in other neurodegenerative diseases such as ALS, motor neuron disease, dementia, including Alzheimer's disease. Aphasia is where people can form words in their brains, but they can't speak it, so that's a communication issue. I showed you data on stroke disability, multiple sclerosis, traumatic brain injuries, and PTSD, something very important to military people we've talked to. So we hope to have a number of studies underway. These are all going to be done with intranasal delivery. We'll keep everyone posted on when these studies will get underway. Now I want to call back my good colleague, Dan Montano. He's going to address just some final issues concerning these medical research studies.
Thank you, Jack. And I've seen that presentation several times, and I have to tell you, both with Daniel and Becky, each time I get emotional when they tell us how they finally have hope and they're seeing progress. So
yeah,
one of the questions that we always get, and we want to get to it right away, why are the medical research studies so small with only 4 people each? The answer is we're trying to learn or discover what works and what doesn't work. We're going where nobody has ever gone before. No one has ever attempted to do what we're doing. One of the questions that a lot of people ask Jack, and I'm going to answer in advance, is people have, do they have to change their medication? Do they have to get off of this or get off of that? The answer is we don't change anything. Correct, Jack?
Correct.
You stay on whatever you've got and we go forward. But we're trying to discover, should we dose 3 weeks? Should we dose 6 weeks? Should we go higher? Should we go lower? Also, the question has come up with people about, well, how long do I have to take FGF1? Well, just like the skateboarder who damaged his elbows, once it had repaired, that was it. The people that we injected the medicine into the heart, once it had regenerated and was normal again, that was it, nothing more. We believe once we reset the brain with FGF1, then that's probably it for 10, 20, 30 years. We don't know. Depends on how much damage you do to it. So we're doing small tests because we're just trying to stick our toes in the water and learn the best way to go. We have a very limited number of intranasal delivery devices due to the supply chain issue. We have very few of the devices that you saw, and therefore we have to be very, very particular on how we're going to do that. At this time, we're only cleared in the British Virgin Islands to conduct medical research studies. We are seeking and attempting to add additional clinics. As Jack said, Bahamas, Cayman Islands, Panama, Morocco, Albania, Dubai, etc. We're out there trying. But once again, we are looking for areas where medical research studies are allowed versus we know the process in the US. We know the US FDA process. We've been there with a heart drug now for almost 15 years. In the United States, it takes 15 years for a drug to be approved. In the British Virgin Islands, it took us 90 days. In Panama, we're starting now, I think, the 5th month, but we think we'll get approved next month. So we're in these very small places, these very particular places, because we want to discover what works and what doesn't work. It is reported that 20 million people worldwide suffer with Parkinson's disease. Zhittya will be lucky this year, that's 2022, to treat 40 people in our medical research studies. That's probably the most we're going to be able to do. 2023, next year, we're hoping to maybe do 180 people, about 15 a month. That's out of 20 million people who want to be treated. The medical research studies are not the clinical trials for approval. They're to help us understand where we are. Remember, discovery means knowing something others don't know. And discovery doesn't mean that you're right in the beginning. Columbus thought he was in India. He was wrong. We're making small discoveries and we're moving forward. If we continue to have success and we find the best dosage level, I then think in 2 or 3 years from now, we can put together a US FDA phase 1 that we're confident will be past the phase 1, which is safety. I feel we have enough evidence now, but not the way the FDA responds. After we get that phase 1, I think in 3 to 4 years from now, we'll be able to file a phase 2. And I think the data will be as the patients improve. From what I've seen already, I think the answer will be yes. So 4 to 6 years from now, I think we can start a phase 3 and be looking to get approval in the United States in 5 to 7 years, if all goes well. But I can tell you in the 24 years that Jack and I have been doing this, it doesn't always go well. So maybe 8, 9, 10 years from now, we will have US FDA approval for our treatment once we know how it works. We already have people coming to us who are saying, I want a medical research study for stroke recovery. I want to be able for dementia, for traumatic brain injury, ALS, multiple sclerosis, and more. But once again, we're limited by our capacity and the devices. We're having to cherry pick what small trials we do to find. Now, as Jack mentioned, we want to do a lot of Parkinson's subsets. But the other reality is we'd love to do a trial on stroke recovery simply so we can get 4 patients and have some evidence if we're right or we're wrong. So if you want to contact us about participating in the research studies, you can go to our website, zgm.care, and click on the link to Cross-Border Medical Tourism. This is a company which we have passed over the entire function of handling our medical research studies and our clinical trials. We have been inundated with people, 30, 50 a day, who want to be in the trial, have questions about this, have this or that, want to argue us about what their neurologist said, et cetera, and et cetera. We can't handle that. We can't go forward and try to come up with a cure for 200 million people while we're busy trying to have somebody tell us that their neurologist thinks we're frauds and crooks. And I still don't understand why people feel they have to call and tell us that their neurologist has no idea of what we've discovered, and therefore there's something wrong with us. But that's another topic. So for information, you can go to our website, zgm.care. Now, Jack, we've got a couple of questions that have come. We had a lot of questions come in, but I'm going to start off with the number one question we've been getting for 20 years, Jack. Does the growth factor cause cancer? Is muscle growth out of control? Is this going to fix my Parkinson's but give me terminal cancer?
Yeah, we've looked hard at that question. It comes up all the time, and we've never seen any evidence of cancer, either in animals that get up to 1,000 times more than we're giving patients, or in our patients. So in those heart studies, FDA made us follow tumor markers. They stayed nice and level. So we've never seen any hint of starting cancer. Now, we would not put someone who has an active malignancy into a clinical trial, but FGF1 does not cause cancer.
Jack, number 2, I sort of jumped into this one, Jack, but for people that were in the medical research study or people that may be considered in the future, will they be allowed to take their regular medicines, or will you take them off of them?
No, they're actually required to stay on their current medicines. What we do ask them is that they don't change something while they're in the research study. So they don't start taking a new Parkinson's disease medication or change the doses. So we want to keep that a level playing field so we're not changing a bunch of things at once. So that's the only stipulation we ask the participating subjects to abide by.
So let me also clarify, Jack. I believe Becky took no Parkinson's medicines, and I believe Daniel did. We did not have them change anything, correct?
Correct. That is correct.
The only thing I'm aware of that changed with Becky was she got a lot more chocolate milkshakes with us.
Yeah, that's true. That's true.
Another question here, Jack, and this one you're going to love. If I'm in a medical research study and you dose me too little and I do not fully recover, then you learn later the best dosing level is level 5 for 30 days. What happens to me? After I help you discover what doesn't work, am I left to suffer after you find out what the correct treatment is?
Yeah, good question. And we will not leave these people high and dry as part of the written contract we'll have with them. They'll be guaranteed any future better dosing regimen that we find for their particular disease. So if we're treating Parkinson's patients and the new dosing regimen is 50% better, those patients will get it. If it's 2% better, I think we probably would think about whether we would retreat those people. But no, we're not going to withhold better dosing regimens from our pioneers, the people who go first, the people who are willing to step up and do things now.
And let me just amplify something. We're not big pharma. Jack and I are in the field. Jack and I met with Daniel and Becky. We understand. We learn from them a lot of things that we never thought about. We are there and we're trying to figure out what is the best way to advance taking care of these people. Jack and I have made the commitment to know the people, and therefore we are fighting for them, not because Jack wants a vacation or a 300-foot yacht because he gets seasick. But here's another question from somebody. I believe you got a spectacular future. I'm interested in investing in your company. How do I do that? We don't discuss investing or anything like that in public. If you're interested in learning more, it's a private conversation. You can email me at my personal email, which is dan333@aol.com. So if you have anything to do with investments, then that would be me you contact. Jack, we have another question here, and I think I semi-answered it, but let me try it on you. When do you think you'll have FDA approval?
I think you answered that quite well. It's going to take 5 to 7 years. We got to go through the animal studies and then phase 1, phase 2. Maybe we'll get a breakthrough designation, maybe not, but we're hoping with all this data we generate outside the US that they may finally let us proceed in the US without killing 40 beagles or 500 rats. So getting all this human data outside the US is helpful, and all the safety data, I think, and having Parkinson's disease support groups say, let these guys try it now in the US.
Let me just tell you a quick story. Under the right to try, we tried to make our medicine available in the United States to an ALS sufferer who had nothing to lose. The person that was sponsoring it was a relative who was a doctor who owned hospitals, whom we had the highest respect for. The FDA turned him down. He wrote an appeal letter saying, yes, we don't know the adverse events of FGF1 in the brain, but we do know that her ALS is going to give her a torturous, painful life. She wants to try, let her try. They turned him down. So when Jack makes his comment that I think that was a breaking point for us, we finally said, hey, listen, if the FDA wants to take 7 years to study somebody who's got a 2-year life expectancy, we got to go find places that are a little more humane than the US bureaucracy. Jack, I got another question here for you. Do you think your treatment would work for progressive supranuclear palsy, Parkinson's disease?
Yes, but yeah, that's a good question. That's, that mimics Parkinson's, the PSP. It's affects different neurons, but it is a motor disorder. And neurons are dying. They're dying in different areas of the brain, like the cerebellum and brainstem. So if that's due to a lack of blood flow, like we believe in Parkinson's disease, then that should be amenable to treatment with intranasal FGF1, and we would certainly consider treating a cohort of PSP patients.
Can I also say something, Jack? We have a lot of people who've asked questions like, how does this work for stroke recovery? How does this work for this? How does it work for that? I'm going to make the big scientific answer here, if you don't mind. We don't know. That's why it's called medical discovery. The first thing I can say is we believe it's safe, and that has been demonstrated now. 8 people have demonstrated they had 0 adverse events. Now, does that mean we can try it on a stroke person? I think more so. But we don't have answers, people. That's why it's called medical discovery. We have to put our toes in the water. We have to slowly search to find out where we go and how we do it. A lot of people keep asking us about the US FDA and clinical trials. that is not a topic you want to hear about. Now, for those of you who are new to us, we have a monthly letter update that goes out. People tell us they're brilliant, goes out to almost 100,000 people now. Go to our website, sign up for the updates. We have many comments in there about the FDA and the approval process. I'm sure you won't be surprised at how candid we are. So, Jack, any other things before we say goodnight?
No, I'm fine. Thank you. Thank you, viewers, for attending the webinar. Appreciate it.
Ladies and gentlemen, we want to thank you once again. If you want more information, zgm.care. We want to thank you. This video will be cleaned up and put up on YouTube, I hope, in a couple of days. And we'll be sending out a notice to every one of you so you can share it with others. We're looking for friends. We're looking for allies. As Jack said, we want to be in a situation that when we take the information that we've gathered from the British Virgin Islands, Bahamas, Cayman, Panama, Albania, Morocco, wherever, and we go to the FDA and say, look, we've dosed all these people. It's been safe. Look, whether you believe it or not, humans are a higher species than mice. We can go to the FDA and say, look at what we've got. We have a group of supporters. We have Parkinson's groups that understand. We give them hope. We give them treatment. We need public support. We know that if we're going to get through the FDA. It is a political organization. It is not a scientific organization. We welcome you. Our monthly updates, I think, are enlightening. Jack writes the best half, and I write the other half. Ladies and gentlemen, thank you for being here. God bless you. And I hope we're right. And I hope someday soon you'll be able to go from having hope to being treated. Thank you. Good day.